Cool. I think we're live. Hi, everyone, and welcome to Neural Launchpad Season 1, Session number 9. My name is Alex Vaccaro. I am a postdoc at Harvard Medical School, and I'm also one of the Neural Launchpad co-organizers. I will be your host for today's session, where you'll hear the work from two very talented trainees. They'll be introduced very shortly by our faculty moderator, Dr. Harrison Gable, who is an assistant professor in the Department of Neuroscience at Washington University in St. Louis. And Dr. Gable will get some help from a graduate student, Urann Chan, who is also from Washington University. And so they'll both handle the Q&A and discussion at the end of each talk and help, the discussion go smoothly. I think we're gonna start pretty much right now. I'll just say two things. So please feel free to ask your questions from on the chat and the ask a question function of Crowdcast. And the second thing is that we have a listserv, and so if you want to register, we usually send an email, one email per week. We promise we're not going to spam your inbox. It's just to tell you, give you the info about the week session and any upcoming season or anything, linked to that, but it's really not to spam your inbox. So again, feel free to sign up. We'll put the link, I think it's already in the chat, actually. I see the link in the chat. So I'm done. Dr. Gable, the stage is all yours.
Great, thank you very much, and thank you for the opportunity to be a host here. It's really a great series that you've produced, and hopefully it's one of those things that, if not created because of COVID, at least is popularized because of COVID. So a silver lining, but a great format. So congratulations. So I'm very happy to host and very happy to introduce two speakers that I know well. I'll get right to it. So the first speaker for today is going to be Gabriella Boulting from Harvard Medical School. It's my pleasure to introduce her. Gabriella received her undergraduate degree in biochemistry from UCLA before attending Harvard University for a PhD, where she worked in the laboratory of Kevin Egan. And for her PhD research, she really pioneered the use of standardized human IPS models to study neuronal function and dysfunction in disease at a time when the field needed to standardize these approaches in order to make insights. And that was really sort of a seminal contribution to the use of these models. And then she joined Michael Greenberg's lab at Harvard Medical School, where I had the pleasure to overlap with her. And this is the first time I've been able to see her in a while. So I'm very happy to see you, Gabriella. And in Mike's lab, she's been studying human-specific gene regulatory programs, specifically stimulus-dependent gene expression programs, and to understand how human and primate-specific regulatory pathways may be at work to modify this pathway and see what it does in humans as opposed to rodent models. And her studies have identified primate-specific regulatory elements, including for the osteocrine gene, which in a recent study showed, and I believe she's going to talk about today, how stimulus-specific expression can be different in human cells, and that can affect dendritic development. And further, she's developing human IPS models to study these mechanisms in specific cell types, and that will be the topic today. So Gabriella, take it away. We're looking forward to your talk.
Thank you so much, Harrison, for that very generous introduction. Okay, I'm going to try and share my screen here, and I might have, oops. Okay, can you guys hear me and see my PowerPoint? Okay, great. First of all, just want to say thank you, everyone, for having me. It's really exciting for me to be able to share my work with everyone. I guess Harrison already gave a great introduction to what I'm going to talk about, the title being Human Neuronal Activity-Dependent Gene Regulation in Development and Disease, because I've been studying in the Greenberg lab what genes are activated and transcribed in response to neuronal activity or brain activity, and what are the non-coding DNA sequences that regulate those genes' transcription, specifically how this is different in humans and primates, and what that might tell us about what's different about our brain's development and potentially our propensity towards disease. And I also like to throw up this alternate title, which is what makes us human, because personally, this is really what drives and motivates me and inspires my research. And although, of course, this is going to be a long-standing question for many years to come, both at the biological and the, I guess, philosophical level, it's really interesting in the past couple decades, some neurobiological, I guess, at least observations have been made to begin chipping away at defining what makes us human, and at least when we're looking at our brains. So, for example, one of the most obvious differences about what's unique about humans and primates in general is the size of our brains compared to rodent models who we diverged from about 80 million years ago. And these are some seminal studies from Herculano Hausel lab as well as the Koss lab that really showed that not only do we have much larger brains and denser parts of our brains, so we have nearly 90 billion neurons compared to 1 or 200 million in rodents, but also there's been expansion of certain sub-regions of our brain, specifically the neocortex, most prominently the neocortex. And that means that in primates, but also especially in humans, we have much greater computing power and new developmental stages of neurons that populate this region, as well as new cell types, even in the adults. And that's been shown and increasingly being shown by multiple labs, from Chris Walsh to Pascal Rakic to Dane Geschwin and the work of Genevieve Konopka and many others. So this is an exciting and continually growing body of literature. And also with new technologies such as diffusion tensor imaging, we're also able to appreciate now there's multiple new kinds of connections being formed in this larger, both within the larger neocortex, but also between neocortex and subcortical regions. But I'd like to argue that it's not just what kinds of cells or what kinds of connections are being made, but also at what rate and what are the molecular processes that are responsible for making those connections. Because something else we know about the human brain is that it actually matures at a much slower rate and then over a much longer period of time compared to other animals. So the process of neuronal birth and migration and synapse formation and then pruning and really maturation overall of the circuitry can just take a couple months in rodents and probably years in monkey. And this is from important work by Jennifer Lund's lab, as well as Rakic lab and others looking in human, where it actually takes probably up to 3 decades. So what is going on during this really long period of time? Well, beyond just our brain growing and these new connections being formed, there are also many different adaptations. And that's what really interests me is, as far as what makes us human, I'm interested in how our genome encodes our brain. So how do our evolutionarily divergent DNA sequences help instruct our brain's development, how they allow our brain to respond, to adapt, to be shaped by our environment in species and primate-specific ways. And as neurobiologists, we all know that a lot of this has to do with several neuronal activity-dependent processes that are always at play, both in development and during adult in processes like learning and memory. So this is a cartoon of a brain cell, of a neuron, with the soma here with its nucleus and one of its dendrites and a spine. And we know that when there's neuronal activity, so communication between neurons, that can cause calcium to increase in the cytoplasm in the postsynaptic cell. And that can cause all kinds of local effects, right? From time spans from seconds to minutes, there's receptor internalization, there's local mRNA translation, cytoskeletal rearrangement. All of this helps the neuron adapt in the short term to the activity in its circuit. But if there's also enough calcium let into the cell that it's actually sensed within the cell body, then that can trigger de novo gene expression, and new gene products can be made and then shuttled out to different parts of the cell to help the neuron adapt to the activity in the circuit in timescales of hours to days. And since I'm really interested in how our genome encodes for our unique adaptive abilities of the human brain, it's really important for me to understand what these what these genes are and also what the non-coding regulatory elements are that control them. And something that I'm really lucky to have is the now decades nearly of research in rodents on what these gene programs are, regulatory programs are, because what we've learned is that in fact, in rodents, it's clear that these gene expression programs in response to brain activity are actually quite cell type specific. And depending on the different cell type or neuronal type that you are looking in, of course, they have unique functions within their circuit. And so they have to adapt to that activity in unique ways. And also, this gave us a pretty good sense that there's likely to be some species-specific aspects to it also. And that's what the hypothesis was of my work to begin with, and it still is going forward, that there are primate-specific aspects of neural development and function that are controlled by unique targets of the experience-dependent gene expression program. So in order for me to understand what this gene expression program is in human and to compare it with what we already knew about rodents, I needed human neurons, and I needed to be able to control their activity state so I can compare transcriptionally across states. And so I was so lucky to join the Greenberg lab and start working with an amazing postdoc, Bulent Ataman, who had established this culture system of human fetal brain samples which were dissociated and plated down in culture, and we could either leave them unstimulated or depolarize these cultures with potassium chloride, which causes synchronous membrane depolarization and robustly induces calcium and activity-dependent gene expression. We did this for different time points and then just compared these different time points by total RNA sequencing, so looking at all the transcripts, all the mRNA in the cells. And at 1 hour compared to 0 hour, we saw several genes induced, as likewise at 6 hours after stimulation. And in black, I have all the genes here, or several of the genes here, that we would expect to see based on our previous studies in rodent. But in blue, we also see genes that had not been previously described to be part of this gene expression program. And CNF331, for example, and Link473. These had not been described in rodent activity-dependent gene expression programs because these sequences are actually unique to the primate genome. So these have newly evolved to be part of this gene expression program, and those are really interesting to look at. But this third gene, osteochrin, was of particular interest to us, not only because it was the most highly induced in these fetal brain cultures, but also because it's a secreted protein. So that really means it has the ability to potentially communicate between neurons in the human developing brain in new ways, much like potentially BDNF and its neurotrophic functions, or potentially some kind of neuromodulatory function. So this is really exciting for us to think about all the possibilities. And the other interesting thing about osteochrin is that its coding sequence is actually conserved among mammals, and it is expressed in the bone and the muscle of rodents, but not in the brain. So we were really interested to think about how could, over evolutionary time, how could this gene have evolved new regulatory mechanisms to now be expressed and induced specifically in the human brain. And after several promoter bashing experiments that I can't go into today, unfortunately, I'll just tell you that we came to find this short 85 base pair enhancer sequence, this non-coding DNA sequence upstream of the osteochrin gene in human, where we identified some 10 base pair stretch that is similar to the sequence known to be bound by the developmentally really important calcium-dependent transcription factor, MEF2. And upstream of that, we found 2 additional sequences that could likely be also MEF2 binding sequences. But those sequences and the critical aspects of them did not seem to be conserved in their sequence in rodents and many other mammals. And so we thought, well, potentially these are 3 MEF2 binding sequences that are only conserved or have only evolved in anthropoid primates. And we did in fact confirm by chip sequencing directly testing the protein binding of MEF2 at this DNA sequence in human, and it was not present in mouse neurons. So to understand if this was true in vivo, we also collaborated with Margaret Livingston to look in macaque to see if using a classic sensory stimulus dependent assay for function in the primary visual cortex, using tetrodotoxin injection in one eye, so this is called a monocular inactivation, could we see down in the primary visual cortex where these inputs are separated just in spatially, still with active and inactive columns called ocular dominance columns, could we see osteochrin specifically only expressed in the active columns, meaning that its expression in the adult primate would be dependent on sensory stimulation. And in fact, that's exactly what we saw. So we saw alternating ocular dominance columns of osteochrin enrichment or expression enrichment by fluorescent in situ hybridization. So here we have an active, inactive, active, inactive columns of expression. And this is different from what we saw, the pattern of expression that we saw of other transcripts, such as at link 473, which I talked about earlier, which is also highly induced in primate and human neurons in response to neuronal activity, but its expression is not restricted to what is layer 4 in the primary visual cortex here. It seems to be represented across all layers of the visual cortex. So we thought this was really amazing, and it's interesting to think about how a certain gene that's conserved among mammals, but not expressed in the brain as far as we know in rodents, can then evolve new regulatory mechanisms to not only be expressed within a specific subtype of neurons in the human and the primate brain, but also in response to activity. And something I can't show you today is our preliminary studies in primary human fetal brain cultures showing that overexpression of osteochrin actually restricts dendritic arborization, and knockdown actually allows for exuberant outgrowth of dendritic arborization. So what does that mean? That means that potentially in the context of this different cytoarchitectural milieu of layer 4, that is quite different from the layer 4 of the visual cortex in mouse, in macaque and human, that neurons need a new mechanism by which to restrict their dendritic field and potentially their synaptic field to promote specific or precise wiring of the visual circuit, and potentially osteochrin mediates that. It's something I definitely want to look into more going forward in adult macaque as well as in human stem cell derived neurons. So overall, what osteochrin has taught us is that it's important to look in primate tissue to understand what the species-specific gene expression patterns are, that very small non-coding changes in regulatory sequences can dramatically alter gene expression, and that we need to understand more about non-coding regulatory elements. And to do that, we need to move to some alternate platforms where we have more homogeneous access to large quantities of isogenic neurons, and namely stem cell derived neurons. To do that, we used actually GABAergic stem cell derived neurons, where we were able to do many more stimulation time points across many more modalities. And just in the interest of time, I'll say that we were able to look at just histone modifications. So what are all the different kinds of enhancers and promoters across the entire genome that are active and activated by calcium signaling, as well as specific binding regions of certain calcium-dependent transcription factors. And this allowed us to not only look at genome-wide patterns of disease heritability enrichment. For example, ASD seems to be specifically enriched in inducible promoters and not enhancers or constitutive promoters of these GABAergic neurons, as well as specific genes, autism spectrum disorder associated genes such as DHR7, which had not been previously described to be regulated by this pathway. But we now not only know about this new gene regulation pattern, but also expect specifically which regulatory elements, which transcription factors, and which sequences that might have evolved to allow for it. So overall, I'm excited that human neuronal activity-dependent gene expression can be studied to identify these novel aspects of our brain's development. And I'm looking forward to continue dissecting and discovering it using both or all three primary human tissues, stem cell derived models, as well as functional validation in adult adaptive processes in the primate. And in the interest of time, I guess I'll just stop here and thank everyone who I've collaborated with. This would not have been possible without an amazing team and a very supportive PI, as well as all of my collaborators, particularly Margaret Livingston, Max Sherman, future collaborators Jason Buenrostro, and several others within the department at MIT and at Boston Children's Hospital. So thank you all so much.
Great, thank you, Gabriella. A great talk. I got a comment from a former Greenberg labber. It was like being in lab meeting again. It's a great.
On fast forward.
So I had a couple questions. How I don't know. I can let you ask a
Sure.
one, and then please post questions in the Q&A down below. So we have. Yep, actually, and this was my question as well. So maybe we'll go to that one first, and then how you can follow on. Does that sound good? So how is this expression specific to V1 layer 4? Are there other genes under its regulatory control? I was interested in that too. Do you have a hypothesis for what's driving, osteochondritis?
Yeah, so
only in layer
I'm really
4?
interested in that. We haven't been able to delve into this as much as we did with the activity-dependent transcriptional control, but there seems to be an evolved sequence much more proximal to the actual transcription start site, which is a long AT stretch, which is actually shortened quite a bit in human compared to rodent, and also because we know that osteochondrin is dependent on co-expression of, well, we've observed that osteochondrin is only ever expressed when the neuron also expresses SAPB2. I think that SAPB2 could be a really important defining factor for either the repression or the activation of this gene, since it can be both a repressor and an activator. And so either setting up that promoter or that enhancer for its baseline expression or something. So I think there are several sequences in the locus that are worth exploring, and also co-regulated transcription factors that are really interesting to look at.
Great.
Gabriella, I also have a question.
Okay, so I'm not seeing other questions in the question box.
Please.
But maybe I can ask a question. Thanks. So I really like the title, the alternative title you give, what you make us unique. I cannot help thinking whether or not all sorts of changes, evolutionary changes that you show, is unique or specific to human or the evolutionary trajectory towards human. What if, for example, if we look at some of the lower mechanism like Drosophila or C. elegans, how do you think that evolutionary transition between those organisms to rodents compared to rodents to us? Do you think there's a steady transitioning, or there's really some abrupt jump towards
I
human?
think that it's been shown also by evolutionary geneticists that there seems to be a continuous genetic evolution, not only between species, but also from our anthropoid ancestors towards hominids and now into modern hominids. So it's unlikely to be an exuberant increase or big, dramatic, large volume change in the number of base pairs in our genomes that have caused like something really human specific and we're special. I think that, of course, just as we found human specific, or we found transcriptional patterns that were in humans that weren't in mouse, there are also those that are in mouse that are not in human. And I'm sure you would see that when you compare between any two species. So of course, remember, every species is perfectly evolved for its ecological niche, and that requires a lot of fine tuning at the DNA and expression regulatory level for every species, and understanding how that happens just generally, regardless if it's human specific or not, is really interesting. I'm not sure if that got to your question, but yeah.
Yeah, thanks. There's another question from Dr. Ann West. And she said, "Hi, Gabriella, love it. One of the most interesting things from the osteochondrin studies is that actually there are not that many differences in inducible genes in general. That is the outlier, but one idea in human is that there may be more cell types than in rodents, and given that much activity-dependent gene expression is cell type specific, do you think that might be something that is worth following up?"
Absolutely. There's, from the human fetal brain culture comparison to mouse fetal cultures, we didn't see dozens of seemingly human-specific transcripts pop up, but a lot of that had to do with how heterogeneous the tissues are. And so we can't really, I think with the upcoming technologies, single-cell sequencing technologies, where you can really look not only within individual cell types and individual transcriptional states, but over developmental stages. And hopefully as the comparative analysis suites get better, when you can compare across species, which is, by the way, still quite difficult, even from the basic gene annotation to annotation comparison level, as those things get better, we're going to really be able to pull out many more differences. And I think at this point, it's just a sensitivity and a shortcoming in our tools to be able to find the large number of differences that I believe are there. But definitely, it certainly depends on the cell type, and it certainly will depend on the developmental stage. And those, we know, not only are there reduced cell types in human in and outside of the cortex, but also new developmental stages. So that's going to be really important.
Great.
All right, maybe time for one more question, then we should transition.
Yeah, there is one more question in.
One or two more. So
yeah. So the last question is the activity-dependent transcription developmentally regulated. Do we have to expect the same gene to be regulated in fetal neurons and in adult neurons?
We should not necessarily expect that any gene is regulated the same way at different developmental stages. So certainly we see from the fetal transcriptome of human brain, so it's such in the brainspan atlas, that osteochrin is most highly expressed in the brain around late gestation and perinatal stages, and seems to taper off after that. And we don't know if it's because the samples that are obtained during adulthood are actually not stimulated, or there's long postmortem intervals, or if that is actually a feature of its gene regulatory evolution or the developmental regulation. And also single-cell sequencing studies have also shown that it can be in other animals as well as human, that it can be expressed in the adult in parvalbumin neurons, interneurons, which is really interesting to think about how a gene's expression can switch between cell types across development. So you shouldn't, I guess, what I've learned is you should just never take anything for granted.
Lots more to study, right?
Always, always.
Cool. Well, we'll keep moving here just to give Urann the next chunk, but thank you for a great talk. And
thank you.
Okay, on to the next speaker. So our next speaker is Urann Chan. He is from Ann West Laboratory at Duke University. Urann received his undergraduate degree in neuroscience with high honors from Brandeis, and then he started his PhD in Ann's lab at Duke. And for his dissertation work, he's been investigating the role of epigenetic mechanisms in synapse development and nervous system function. And he's going to be telling us today about his studies to define the repressive role of the histone H3 lysine 27 methylation mark in the regulation of gene expression in cerebellar granule neurons, and describing how disruption of this methyl mark impacts regulation to affect neuronal function and ultimately modify behavior. So take it away, Urann.
Thank you. Can you hear me?
Yes.
Okay, perfect. Can you see the slideshow?
Yep. Yeah, for anyone who can't see it, I assume everyone figured out if you double click, it comes up to the top. But
all right. So thank you, Harrison, for that introduction. So today I'm going to be telling you guys about a project that I've been working on. I'm going to tell you guys about a project that I've been working on for one of my thesis projects, and that's investigating the role of chromatin regulation in synapse maturation and social behavior. Now, whether you're looking at the human brain as illustrated here, or the mouse brain, we know that brain development and maturation are long-lasting events that begins during embryonic development, goes through birth, and extends throughout the life of the animal. And during this time, the brain has to integrate a combination of both intrinsic and extrinsic cues in order to regulate behavior. And so to do so, the brain has to have the proper connections and wire accordingly. And so it's imperative that we study mechanisms of brain development in order to understand how those connections are established. And so in the West lab, we utilized the mouse cerebellum as a model system to study brain development. And the reason why we use the cerebellum is because one, a majority of the cerebellum develops postnatally, which means we can manipulate the system after birth. And second, the cerebellum provides a very tractable system to study the development of a single cell type, the cerebellar granule neuron, which I'm showing here, as it goes through the different stages of development, starting with proliferation of the progenitor pool that starts during the end of embryonic development, through migration, dendritogenesis, and then finally synapse maturation that happens all within about the first 3 weeks of postnatal life. And then during this time, there are dynamic changes in gene expression that occurs that results in the expression of different markers that really characterizes the developmental state of the cells that are present during that time. So here are sagittal sections from postnatal day 7, 14, and 60 animals. And I'm showing you in P7 animals, the cerebellum is enriched for Ki67, a marker for actively dividing cells when the progenitors are present. At 14, it's enriched for double cortin, a marker for migrating in newborn neurons. And then P60 is enriched for the alpha-6 subunit of the GABA-A receptor, which is highly expressed in the cerebellum. And so what this tells us is that in order to understand brain development, we have to really understand how the brain orchestrates this fine balance between gene regulation, the expression of genes to encode for products that help to set up the circuit, and ultimately how those circuits mediate behavior. And so for the purpose of my talk today, I will be focusing on the latter two, really asking what is the role of Kdm6b, my gene of interest, in cerebellar development, as well as its role in social behavior. So what is Kdm6b? Kdm6b is a lysine-specific histone demethylase that removes the repressive chromatin mark H3K27 trimethylation. And so during development, methylation is placed on this on lysine 27 by a repressive complex. This in turn condenses the nucleosome and puts all nearby genes in a repressed state. Kdm6b is able to remove and catalyze that repressive mark, thereby decondensing the nucleosome, thereby putting nearby genes in a more activating state. And so a couple of years ago, my lab was really interested in understanding or trying to figure out the genes that were regulated by Kdm6b in the cerebellum. And so what we did was we cultured cerebellar granule neurons from P7 wild type animals, and then infected the neurons with lentivirus encoding shRNAs targeting Kdm6b to really ask what are the genes regulated by this demethylase. And we found that the majority of the genes that were regulated were genes that normally turned on later in time, which we dubbed a mature gene set, or genes were important for maturation. And when you lose Kdm6b expression, the expression of these genes downregulate, suggesting that under normal conditions, Kdm6b works to upregulate the expression of these genes over time. And so when I joined the lab, I was really interested in studying synapses because that's what I was studying that when I was an undergrad. And so when I looked at these gene sets, I noticed that a portion of them encoded for synaptic proteins, genes like DACT1, GRIN2C, SLC17A7, and Wnt7A. And so I made this simple hypothesis that if Kdm6b regulates the expression of synaptic genes, then it has to be required for synapse development. And that was really the question, the driving question that kickstarted my thesis. And so in order for me to visualize synapse development in vivo, I utilize a technique called an in vivo postnatal cerebellar electroporation. And this technique is actually quite advantageous because one, it allows me to sparsely label the cells and do three-dimensional reconstruction, which I've shown here as a three-dimensional trace. And additionally, it also allows me to genetically manipulate the cell and then study that manipulation in a cell-autonomous manner because I'm only targeting a very small population of neurons. And so in order to ask the role of Kdm6b in synapse development, what I did was I took P7 homozygous Kdm6b floxed animals. I electroporated either a GFP cell fill as a control or the same GFP cell fill, but with a construct expressing a nuclear Cre recombinase to excise the region flanked by the LOXP site and knock out Kdm6b. I then waited 7, 10, 14, and 23 days post-injection before looking at morphology. And then specifically, I was interested in these specialized structures called dendritic claws. They are synonymous to cortical spines in that they're where a majority of the synaptic inputs onto granule cells are located. And so I was really interested in seeing their development. And if you look at a control cell over time, you notice that as the cells develop and as their synapses mature and refine, the claws actually grow in size and become enriched with synaptic protein. And so the first thing I wanted to measure was the neurite shaft, basically the length from the soma of the cell to the base of the claw. And I found no significant difference between either controlled cells or Kdm6b knockout cells. However, when we look at the actual length or complexity of the dendritic claw, the knockout neurons or neurons lacking Kdm6b have a significant reduction in claw size as the claws begin to grow during a time when synaptogenesis is really critical. And if you look at the representative image at 23 days post-injection, you'll notice that the knockout neurons or the neurons without Kdm6b are about a third smaller in size. And to show that this effect was specifically due to loss of Kdm6b, I performed a rescue experiment in which I knocked out the endogenous Kdm6b and re-expressed a human Kdm6b construct. And although this is a little bit preliminary, basically what it shows is that electroporation of the rescue does cause the claws to sort of return to baseline control or similar to the control. And so the next question I really wanted to ask was, now that we know that Kdm6b affects the size of these claws, there's essentially less real estate for synapses to form, being that the claws are where the synapses form. And so I wondered if Kdm6b controlled claw formation, can loss of Kdm6b result in defects in synapses as well? And so here I'm showing you a schematic, which is a close-up of a CGN dendritic claw. And during development, mossy fiber terminals from places like the pontine nuclei innervate the granule cell layer. And the dendritic claws of these CGNs will actually synapse onto these mossy fibers, forming what's known as the cerebellar glomerulus. And it's the co-localization of presynaptic VGLU2 from these mossy fibers and the co-localization of postsynaptic PSD95 that really signifies a morphologically mature synapse. And you can see this in the maximum projection image with the same color scheme that I've highlighted in my drawing. And so when I analyzed PSD95 puncta on these dendritic claws, I noticed something really surprising. The knockout actually have a higher density of synaptic proteins on their claws compared to the control. And I assume that this is most likely due to the fact that the claws are smaller and they're unable to redistribute the synaptic proteins accordingly. And then when I looked at the co-localization of both VGLU2 and PSD95, again, we see that the knockout have a higher density of synapses, but at a much later time point, probably when synapse number are a bit more crystallized in the cerebellum. So taken together, what this shows is that Kdm6b is required for claw formation and required for proper synapse formation. And it could suggest perhaps a failure in, say, homeostatic property in that the cells can't recognize that their claws are smaller and therefore they can't distribute their synapses accordingly. And so this actually raises a really important question, because when you look at the cerebellar circuit, granule cells synapse onto the Purkinje cells. So if we're messing up claw formation and we're messing up how the synapses are distributed, what does that say about the inputs from granule cells to the Purkinje cells, right? And that's something that we have yet to explore. And so that brings me to the end of the first portion of my talk. For the second portion of my talk, I really wanted to talk about the role of Kdm6b in behavior, and specifically social behavior. And I really wanted to ask, can changes in chromatin regulation drive changes in social behavior? And so to do this experiment, we collaborated with Kai-Gi at the NIH. Kai-Gi had made this enzymatically-dead Kdm6b knockout mouse line, enzymatically-dead Kdm6b mouse line, which I will refer to as the Kdm6b knock-in. And so in order to create this mouse line, he CRISPR knock in 5-point mutations into the demethylase domain. And if you look at the animals here, the homozygous knock-in mirrors that of the constitutive Kdm6b knockout, in that the animal is perinatally lethal. So for the purpose of my experiments, I focused on the Kdm6b knock-in heterozygous, which has one enzymatically dead copy of Kdm6b, and then the wild type littermate. And we were really interested in this mouse initially because we noticed that the mutations that were knocked into the demethylase domain were similar to mutations found in patients with autism spectrum disorder. And so these mutations were, again, into the demethylase domain of Kdm6b. And so so when I got these animals, I bred them up, I ran them through a battery of behavioral assays and found the most robust differences in sociability. And so the first test that I wanted to show you is a test known as social recognition and novelty. And if you're unfamiliar with this paradigm, it involves 3 phases. The first phase, I introduced the experimental animal into 2 inanimate objects to basically set a baseline preference. In phase 2, I replaced one of the inanimate objects with a novel social partner to test for social affiliation, whether or not the animal can discriminate between an object and a social partner. And because mice are social creatures, they will interact significantly more with the novel partner. And in phase 3, I replaced the other inanimate object with another novel partner, which means that the partner from phase 2 now becomes the familial partner. And this is to test social novelty, whether the animal can discriminate between a familiar partner and a novel partner. And so here I'm showing you a preference score, which is an indication of how much more time is spent interacting with the novel partner in phases 2 and 3. And wild type is shown in solid, the knock-in is shown in stripes. And in phase 2, when you're looking at whether the animal can discriminate between an animal and an object, both genotypes prefer to interact with the social partner. However, in phase 3, when you're comparing this to baseline, while the wild type prefers to interact more with the novel social partner, the knock-in is unable to really prefer either the familial partner or the novel partner. And this effect is actually more apparent when you look at the normalized interaction time. So in phase 2, both the mutant and the wild type interact significantly more with the social partner over the inanimate object. However, it's only in phase 3, only the wild type is able to interact more with the novel partner over the familial partner, while the knock-in is unable to, or the knock-in prefers to interact with both on a similar time scale, which suggests that they cannot discriminate that this is a novel partner versus a familial partner. Now, before I start sort of making any, conclusions about this very specific phenotype, I really wanted to test the robustness of this effect. And so I ran a different cohort of animals through another paradigm, really trying to tease apart this social novelty defect. And so I ran the animal through what's known as a social habituation paradigm. And in this paradigm, the animal is introduced to a novel social partner. I take that social partner away for about 10 minutes, and then I reintroduce the same social partner in a second trial. This is then repeated 4 times, where I present the same social partner every single time after an interspaced interval of about 10 minutes. And so wild type animals, after seeing the same partner every time, they realize it's the same partner and they become less interested, right? And so the amount of interaction is actually highest in the first trial when the animal is most novel, and it will decrease in each subsequent trial as the animal habituates to the social partner. And it's shown here on this graph. And so based on our last behavioral paradigm, we think that the knock-in animal is unable to discriminate between partners, right? So in theory, they should not be able to recognize that it's the same partner throughout every trial, and they should not be able to habituate across multiple trials. And that's exactly what we see in the data, right? That the knock-in animals fail to habituate. They actually interact more and continue to go on that trajectory. And so this corroborates the previous finding, suggesting that these knock-in animals that contain the demethylase-dead Kdm6b cannot recognize and cannot discriminate between different social partners. All right, and so basically that's the two story I wanted to tell you. So in summary, I've shown you that Kdm6b regulates the expression of synaptic genes in these granule neurons, and we have ongoing experiments now to tease apart whether Kdm6b regulates the expression of these genes through its enzymatic functions or through non-enzymatic means. I've shown you that Kdm6b is both required and sufficient to drive CGN synapse development, and how that interferes with the cerebellar circuit is something that we have yet to explore. And then lastly, I've shown you that constitutive loss of the Kdm6b's catalytic activity results in a social novelty defect. And like I said before, we were really interested in this because these mutations were similar to that found in patients with autism. And so we have ongoing experiments currently that will ask whether ASD-relevant mutations in Kdm6b affects its enzymatic role in catalyzing H3K27 trimethylation. And so with that, I'd like to thank my lab. I'd like to thank Ramona and Chris Means and the Behavioral Core for helping me set up those behaviors, as well as my funding source. And with that, I will take questions.
Okay, great. All right, we got a couple questions showing up here.
Okay. Do you want me to
Sure,
read it?
go ahead.
The first question is the knocking brain-wide? And do you think the effect observed with respect to failure to habituate to a social partner is a learning defect resulting from issues in synapse formation and perhaps synaptic plasticity?
So the knock-in is whole body wide. It's constitutive. So it it's in every single cell. Kdm6b is ubiquitously expressed throughout every cell of the body, and so it's a whole body knockout. And actually, that's a really great question. Whether what do we think the role of this, or do we think that the social defect is dependent on its whether it's a cortical issue, whether it's a cerebellar issue? It's hard to say because it is everywhere, and it's something that we do have to follow up on. Though, if I like to think that it's cerebellar specific, but obviously we don't know that that's the case. But the cerebellum, for example, does project to many areas of the brain. But it's a hard question to answer, I guess.
Great. So the second question is, are there any examples in histology of human neurons of an effect of Kdm6b mutation? For example, the cloud shape?
I'm sorry, what was that question?
Are there any examples in histology of human neurons of an effect of Kdm6b, right, mutation?
Yeah, 6B.
Yes.
Yeah.
I don't think so. I mean, I haven't seen any studies looking at histology from patients with Kdm6b mutations. I know that there are still patients who are, from recent studies with Kdm6b mutations. Some of them do have stunted growth, but not all of them present with stunted growth. So and some of them do have mutations in other parts of Kdm6b that's not the demethylase domain, or they have mutations in other genes. So it's hard to pinpoint whether any brain abnormalities that they have is dependent on Kdm6b. But that being said, if we look at rodent studies, knockout of Kdm6b, either brain-wide or brain-specific knockout of Kdm6b is perinatally lethal. And in that aspect, the cerebellum is actually affected. But specific knockout, like a cerebellar knockout, does not affect cerebellum at all, actually.
So maybe an early role of specification, something like that.
Mm-hmm.
So Gabriella asked a related question to what you were talking about before. I don't know if you want to elaborate. Do you think that the social deficits are likely caused by synaptic deficits within the cerebellum? Sorry, Gabrielle, I should let you ask.
No, I mean, it's, again, a great question, and it's something that Anna and I have talked extensively about trying to figure out. You know, there's a lot of people that's working on how the cerebellum, you know, inputs in the cerebellum, right? Like, you know, someone on my committee member, for example, right? Like Court studying prediction learning in cerebellum. And so it's the hub. It's a hub that projects to many different areas. And so, and, you know, being that granule cells are so numerous, and they project onto the Purkinje cells, which are the main output cells that project outside of the cerebellum, you know, what does that, when we disrupt the synaptic inputs, what does that, how does that affect the, I guess, the integration of activity onto the granule cells, and how does that change, you know, the way that the Purkinje cells read those inputs and obviously act accordingly. And so it's something that we are going to follow up on. I mean, we're probably going to look at Purkinje cell morphology in the knockout, but that's something that I don't have an answer to, unfortunately.
Yeah, it's interesting to also think about the correlation maybe in the patients or in the mice with these specific deficits between motor deficits. If there are any motor deficits, obviously that's the classical understood role of many aspects.
There are no motor deficits. So that's the interesting part. So I ran these animals through a neurophysiological screen. I ran them through I ran them through about 10 different behavioral paradigms. And they're almost normal. They're basically normal in every other aspect, right? They have regular reflexes. They have normal grip strength. They're able to move their limbs. They're not, you know, they don't have a gait. They don't have tremors. They don't have anything like that. And so, you know, it's interesting, right? Like you can get rid of the enzymatic activity of a whole demethylase. And
what about the olfactory? Sorry, what about the olfactory function? I wonder.
We did not actually look at that.
Okay.
Yeah.
Since that's, as I understand, maybe the basis of the social behavior in mouse, in rodents.
So related to social behavior, actually, there's a question of, is there a similar social deficit observed in human ASD subjects? Maybe specifically those with Kdm6b mutations.
Mm-hmm. So a subset of patients with Kdm6b have autism, which is characterized, right? Which one of the characteristics of being autistic is that you have to have social, you have to have social deficits. And whether social novelty is affected, I assume in patients with social deficits, a lot of different types of communications are affected. Whether social novelty specifically is affected, I don't.
Yeah, we tend to have different measures for the mice than for the humans, right?
For humans, exactly.
I wonder, you know, if there's been a number. It's on the Safari Searchlight list, right? Which means it's been identified with de novo mutations. Can you parse the de novo mutations as autism associated versus the ones that are from individuals that were not diagnosed with autism? Is there any clustering of those mutations? Sorry, I jumped in.
They're not, from what I. So the mutations are quite specific. I mean, even, you know, if I remember the study that I referenced in my talk, there would be, you know, they took patients who had both de novo mutations as well as non-de novo mutations, and there was really no correlation to whether a patient, you know, whether de novo mutations resulted in a higher likelihood that they have autism or not.
Yeah, so you can just, there's a higher prevalence of people with mutations in ASD, but when you look at the gene, it doesn't directly parse with phenotypes.
But you are right in the sense that it is pretty high on Safari. It's a category 1 Safari gene. Yeah,
it's a Searchlight gene, so there should be more mutations coming, right, as and workup of individuals, right? So for those of us in the autism world, we can go back to Safari and they should be reporting sort of phenotypic workups, and we might be able to start to parse the types of social deficits and other deficits that the individuals has as those data sets become available. So we'll look forward to that. There was one last question. How do you want to do that
Sure.
one.
This one has been voted twice, which says, great talk. Do you use male and female mice for your studies? If so, have you seen any sex differences?
Yeah, so all of the studies I've done, yes, I use both male and female mice, and the data I showed was a combination. So based on numbers alone, I had more males than females. And if you actually do out the Mendelian genetics, it's, I think it's female knock-ins are like the smallest group. It's slightly a little bit altered. So my data would suggest that the effect is more robust in males, but I think it's because I had more animals in males. But the effect is still present in females.
Great. So yeah, there's no more question in the question box. Thank you.
Well, we're coming.
Yeah, I guess I have one question, and I think it can apply to both of you guys. I was wondering, like, do you or anyone like ever look in the enteric neuronal cells, and the enteric nervous system, and see how actually, like, this is different from neuronal cells in the brain in humans or mice or whatever model they have. And this is a question, I mean, it's a personal interest of mine, so that's why I'm asking, but I always like to hear differences or actually similarities between.
Well, quite relevant for autism,
so,
right?
Yeah.
With gastrointestinal issues being associated with disorder.
Yep.
I know that David Ginty has been looking at some sensation in autism, but I'm not sure if other people in the enteric nervous system world are specifically looking at differences in autism or activity dependence. But I expect that probably anywhere where there's a difference in either formation or function of a circuit, that there is going to necessarily be some differences in gene expression also and regulation. So I'd expect that the more resolution that we have and the more places we look, the more differences we'll probably find.
That's actually, sorry, go ahead, Urann.
No. I was just really gonna agree. I was just gonna agree with Gabriella. I think it's hard to say because that's not my area of expertise, but it's, I don't know, it's definitely something that you want to be, I definitely want to be aware of, especially because I'm working with these animal models where I'm affecting the whole body, right? And of a gene that is expressed everywhere, right? So it's hard to really pinpoint, whether a defect that I've noticed is dependent on one brain area, or whether a different nervous system or or something completely outside of the brain, right? This could all just be something because somewhere, in another brain, in another body region.
Yeah, actually related to that, I was wondering if you plan to use some of the conditional knockout with, the cerebellum Cre to parse things out. I don't know, maybe that is related to your stage in grad school.
Yeah, no, we have already those experiments. So I have the conditional knockout. So it's a Kdm6b flux crossed to an 801 Cre. And I'm in the process of looking at synapse development in that, as well as the knock-in. But surprisingly, I don't expect an actual change in total synapse number, because in my electroporated cells, there's not a drastic change in synapse number. The change in synaptic density is actually purely driven by the fact that the claws are not fully formed.
Mm-hmm. Right? That's a little bit early Cre, right?
Yeah.
Mm-hmm.
So you knock it out early on, and then, but the animals are fine. They live. They have normal developing cerebellum. There's no difference in layer size or cerebellar size or anything like that.
I see.
At least starting from P14, which was the earliest we looked. Perhaps it could be earlier that there's a defect, but.
Mm-hmm.
Great. I think we've finished up the hour, so.
Yeah, I think we made it. Thank you so much to all of you. I want to congratulate our two speakers. You guys gave really great talks. I think it was really interesting. The discussion was also pretty cool, I think. Thank you to our moderators. Thank you for your time. And yeah, I guess we'll see you next week. This will be the last session of this first season. So, you know, same time, same day, Wednesday, 2 p.m. Eastern time. And yeah, like if you missed the beginning of this talk or previous sessions, you can watch the replay. Just go to our website. The link should be there. And yeah, I guess have all a great day and see you soon. Bye.